The screen is visible, say yes or no. Then I'll move forward.
Yes, sir.
Yes. But when we come to oocytes, we were discussing glands before that. So there are 3 glands, and these glands are very important for secreting things or factors which help in 3 things: sperm maturation functionally, the motility of the sperm, and viability of the sperm. Now the term viability means everything clubbed together to make the sperm able to fertilize the ovum. Is it clear? Reem and Madhya?
Yes, sir.
Yes, sir.
So let's first come to seminal vesicle. So quickly, I'll tell you, seminal vesicles make around 60% of the total semen, the fluid we call semen, which are together released by these 3 glands. And the main secretion of seminal vesicle are these 3 things. First is fructose. Okay, you know what is fructose? It is the energy. I think we discussed, I and Reem discussed it in the last class. Remember, Reem? Sperms are the only cells which depend on
Yeah, sir.
for their energy source. So that fructose comes from seminal vesicles. Second thing that secrete is prostaglandins. Okay. By the way, both Reem and Madhya, did you get the links for my recorded lectures, which are the YouTube link? You did, Madhya? You?
Sir, I have to get it. I will get it today.
Yes, please get it today, and please, because those are recorded, so you can go through it any time of the day. Go through it, make the notes, because all of this is discussed in detail, okay, which is not possible for us to discuss in a very short period of time. But let's come to the major focal points. So you have to know that seminal vesicle secretes fructose, something called prostaglandins, which are very important for motility and viability of the sperms, and certain proteins which help in a slight coagulation of the semen. So when semen is released from the male's body outside, when it is ejaculated, post ejaculation, it starts getting a little more thickened, viscous than it was when it was in the body. And that happens because of these coagulation proteins. Is it clear? So all these 3 important things comes from seminal vesicles. Is it clear?
Yes, sir.
Yeah. Then is the prostate gland, which majorly is responsible for secreting the alkaline fluid, milky colored alkaline fluid. It makes the semen alkaline, and the function is basically to provide the basicity. You know that sperms cannot survive acidic environment at all. Both Reem and Madhya, you must have been knowing this, right?
No,
Yes,
sir.
sir.
No, you don't know. So write down: sperms cannot survive acidic conditions. Okay. That's why semen has to be alkaline, so that sperms can be healthy. And that alkaline thing is secreted by prostate gland and bulbourethral gland together. So prostate gland secrete a thin milky colored alkaline fluid, and bulbourethral glands secrete the same alkaline fluid with mucus. What does mucus do? Anywhere in biology, what is the role of mucus? Simple.
Lubrication.
Yes, perfect. All our internal linings, all our gut lining, nasal lining, all these linings are coated with mucus. The stomach is coated with mucus. So one thing is lubrication. Second thing is protection as well, right? Remember, our stomach produces a very strong acid, but still nothing happens to the tissues of the stomach. How? How? Remember class 11 studying digestive system, Reem and Madhya? There is a very thick mucus lining in our stomach. Remember that? Am I audible? First of all, I am audible, right?
Yes, sir.
Yes. So please, say something, because I don't know when if I'm audible or not, if there's a signal distortion. So similarly, mucus here plays first role as lubrication, and these bulbourethral glands, write down if you want to write. You must be knowing that they are the first one that gets activated upon sexual stimulation. So in males, whenever there is sexual stimulation, these glands start releasing this alkaline mucus fluid, which neutralizes the acidity of the urethra. You know, urethra is a common passage for both semen and urine, for the sperms and urine. Yes?
Yes, sir.
And what is the pH of urine? Is it alkaline, acidic, neutral? What is it? Urine.
Alkaline.
No, urine is a waste substance. It has to be acidic, right? So its pH is always acidic. Urine is always acidic. Okay, Madhya and Reem. So now
Yes.
we have a problem. If urethra is a common passage for both sperms and urine, the urge for urination, you must have studied this in excretory system chapter. Then when kidneys filter urine, on an average, every male urinates around 5 to 6 times a day on an average. Yes, which means urination is more frequent than ejaculation. Yes, so most of the time that urethra is acidic, the lining is acidic because it's coated with urine. Yes or no? Internally, correct?
Yes.
So if the reproductive system allows the sperms to just go into that acidic environment, then a lot of sperms will be killed before they are actually sent into the female tract, right? So they are just a waste. Yes or no? In our own system, in the male's own system. Yes.
Yes, sir.
Making sense? Is this discussion making sense to you, to make concepts?
Yes.
Yes.
Yes. So for that, the bulbourethral glands first have to start secreting much before ejaculation, right after a sexual stimulation. When males get into a arousal feeling, the bulbourethral glands start lubricating the whole line and making it alkaline. They are preparing the urethra for an ejaculation. Okay, so they are neutralizing all the acidity and making it alkaline. They also neutralize the secretions from bulbourethral gland are a part of semen, and they also neutralize the vaginal acidity, the acidity which is present in the vaginal tract or the cervix. Okay, and of course it provides lubrication. So protection and lubrication both together. Is it clear? That's the role of mucus everywhere in the body.
Yes, sir.
Perfect. And then about semen, I've already told you that it's 60% is from seminal vesicles. Rest 35% is from prostate glands, and very less is from bulbourethral, but the first one, the very important one, to reduce the acidity. Now, one thing that I want to discuss with you before we join the batch is oogenesis. Now, you know what is oogenesis? Reem and Madhya, you have studied about oogenesis as well in your schools.
Yes, sir. It is the formation of
Yes.
female what is that?
Female gametes.
Yeah, female gametes.
Yes, perfect. And always there comes a comparative analysis of oogenesis with spermatogenesis. So do you know how oogenesis is different from spermatogenesis?
Spermatogenesis is the formation of male gametes.
Is a formation of?
Male gametes.
Yes. And what is oogenesis?
It is the formation of female gametes.
Yeah, but how is it different? Is it both the same? Both are happening through meiosis. Okay. Is there any difference? Yes, there is a difference. How many sperms do male produce and secrete per ejaculation? Any idea? Please allow me to have some hot tea. My throat is not good today. Millions.
20,000. Yeah,
1,000.
millions.
1,000 will not at all work. It's a condition called oligospermia. If a male is secreting sperms in thousands, he has very less chances to be able to fertilize the female. Per ejaculation, on an average, in a normal human, there are 300 to 400 million sperms. Do you understand it? Around 30 crore sperms.
Yes.
It's a lot. And still, after all this huge number, fertilization is a chance event. It may or may not happen. Yes?
Yes.
But how many oocytes do female release every month? Not every day also, every month. In 1 month, one ovary releases only one oocyte. One single precious oocyte for which 300 million sperms are just competing to fertilize. Is it making sense? Is it clear, both of you?
Yes, sir.
Yes.
Now there has to be a difference, because first difference is oogenesis begins before birth. You know that? In embryonic stage. So oogenesis in a female starts before birth in the embryonic stage, whereas spermatogenesis begins after puberty. Okay, in a male testes. Is it clear, everyone? Both of you, Reem and Madhya?
Yes.
Yes.
Second difference is before birth. So there is a very interesting and famous neat question. The question is, the ovum from which you are born, like Reem and Madhya, was produced inside the body of, and mother was not an option. So what will be the right option if there is mother is not there in the option? So when is oocyte?
Grandmother.
Maternal grandmother. Yes. You understand why?
No.
No? Okay. So when your mother was an embryo inside your maternal grandmother's body, then when your mother was developing as an embryo, in the 3rd month of your grandmother's pregnancy, your mother started developing the ovaries, tiny small ovaries, embryonic ovaries, and within those embryonic ovaries the primary oocytes started forming. Is it clear?
Yes, sir.
And then they got stored. So eventually after birth, when your mother was born and then when she reached puberty, after puberty there is no new oogenesis happening in a female ovaries. Everything has already been formed before birth, when your mother was inside your grandmother's womb. Is it clear now? Is it making sense?
Yes.
Yes, sir.
That the ovum from which you were formed was actually produced inside your maternal grandmother's body. So let's come to this flowchart here. Look at it. The primordial germ cell, again it starts with primordial germ cells, and inside the ovary it forms oogonium, just like spermatogonium. Okay, then oogonium forms primary oocyte, just like primary spermatocyte, which is again a diploid cell, just like primary spermatocyte. This primary spermatocyte starts meiosis 1. Is it clear?
Yes.
But
Yes.
it does not complete it. It starts and gets arrested at prophase 1, the first stage itself. Okay, it gets arrested at prophase 1, and then millions of these are formed. Millions, and when I mean millions, it's like around 10 to 20 millions in each ovary. But after that they start declining very quickly. They start dying, which is called the phase of atresia. So they are arrested and now they are dying and they degenerate, and they are arrested at M1, and the birth takes place. So this is where the females are born at this stage. When they are arrested, then the females are born. And from birth to puberty, nothing is happening with respect to oogenesis or maturation of oocytes in the ovary. Is it clear? Only after
Yes, sir.
only after puberty, this meiosis 1, which was arrested, gets completed before the ovulation, and then through menstrual cycle, the first oocyte is released, which is a secondary oocyte. So the thing which is released is not a complete ovum, it is a secondary oocyte. And unlike sperms, remember in case of sperms, I think there is something that you are not able to see here. There is some problem. And so in case of spermatogenesis, it was very straightforward and clear, right? Meiosis 1 happens, 2 secondary spermatids are formed, spermatocytes are formed, and then meiosis 2 happens and 4 spermatids are formed. Yes or no, everyone? It was very simple, right? Here it is not that simple, because ultimately the female system is allowed to only secrete one oocyte. Can you see here? the fully mature ovum. So what it does is, after meiosis 1, it makes only one secondary oocyte, unlike spermatogenesis. And the second cell is a polar body. It is a very thin, very small, discarded cell with just the nucleus and a little bit of cytoplasm, nothing else. Is it clear? Everything comes to this secondary oocyte. So it is called a unequal division. Unequal in case of, in terms of size and resources, but not in terms of nucleus. Nucleus for both is haploid. For the polar body also it's haploid, and for the secondary oocyte also it's haploid. Is it clear to both of you?
Yes,
and then again, when meiosis 2 completes, again 2 cells should be formed from each. So this polar body is unable to divide further. It is gone. It is discarded. Now the secondary oocyte again follows the same rule. Unequal division, meiosis 2, one becomes ovum, a full mature egg with lots of cytoplasm, a haploid nucleus, everything inside it, all the cell organelles, everything. But this polar body again is a discarded restricted cell with just a nucleus and little bit of cytoplasm. Is it clear? So that is the difference between the male and the female gametogenesis. Is it clear, both of you?
Yes, sir.
So when you, what I want for you is, when you read oogenesis from your NCRT, make a parallel flow chart and come up with your doubts. Okay, Madhya and Reem?
Okay, sir.
Okay, so
Okay, and then we'll take that tomorrow. Any doubt that you have in chapter 3, I'll take tomorrow. Okay, everything. So please list
Okay,
all.
sir.
When you will join, we will just keep on going with your doubts and we will see. Okay, we have been joined by Mamuna, Minashi, and Saifullah. How are you, everyone? Am I audible? And,
Yes, sir, fine.
am I audible and visible to all of you?
Yes, sir.
Yeah, okay. So let's quickly, let me just, okay. So I'll share it again. Any doubts? Saifullah, Minashi, Mamuna?
Sir, I was absent the last few classes.
Yes, and why were you absent?
I went to the outside here. I was not able
Okay.
to join.
Okay. Okay, so we'll, so did you go back and read from NCRT? Do you know where we are?
No.
We have actually started genetics. Okay, so what all lectures did you miss?
The
How many
last
lectures did you?
topic, I was in high one.
Okay, so probably you have missed 2 classes, right?
Yes.
Okay, so what I'll suggest is, Saifullah, do one thing. Get the recorded lectures, go through it, and then you can also join tomorrow a little early, half an hour early, with Reem and Madhya with your doubts if you have any. I will be taking doubts from chapter 3 tomorrow, all the doubts. Okay, Reem, Madhya, and Saifullah, will it work for you?
Yeah, okay.
Okay.
Yes, sir.
Okay. So let's start with today's. We started with genetics, right, everyone? Just the basics. I think we did basics in the last class. This is the notebook. Can you see the screen that I'm sharing, everyone?
yes.
Just one moment. Sorry. Let me arrange things. Okay, give a quick recap what we did in the last class, because I think Saifullah was also absent. So Reem, you were there, right? Would you like to give a quick recap to me?
Sir, I left early, right?
Oh yes, yes, you left early. Madhya, you were there?
Yes.
Yes. Would you like to quickly give a quick recap in 5 minutes?
So we started with the genetics chapter, and then we discussed what was the meaning of inheritance and variation. Inheritance means the transfer, the traits or genes or any characters that are passed from one generation to another. And variation means the change that comes in the offspring and they are not similar to the parents. And the inheritance happens through gametes. And then after that, we were discussing about Mendel's law of inheritance, and he used the pea plant for it. And he used the pea plant because of the characters. They were very distinct and multiple distinct characters. And the pea plant also had a shorter life cycle and produced a lot of seeds in a short span of time. And like we could do both self and cross pollination artificially.
Yes. So just some basics that we did in the last class, so no one missed something significant. And then we also discussed about what traits did Mendel chose, right, Madhya?
Yes. And then for the plant height, he chose a tall and a dwarf plant, and the seed shape was round. The dominant one was round and the recessive one was wrinkled. And then the seed color for the dominant plant, it was yellow, and for the other one, it was green. The recessive trait was green. And the pod color was green, and for the dominant trait, and for the recessive one it was yellow. And so basically the ones which we get at home are recessive traits.
Yeah, right.
And
so
then
yes, then continue.
The pod shape we discussed, the dominant one was full and the recessive one was constricted. And for the dominant plant, the color of the flower was violet, and for the recessive one, it was white. And position for the dominant one, the flowers had an axial position, and the recessive trait was terminal.
Yes, correct. So these were the 7 traits, characters that Mendel chose, and this is the dominant and the recessive trait for all. Now, and then I ask you to go back and study about the laws of Mendel, right? I think you have studied it in your lower classes as well, right? Just the basics of it, correct? You remember what are these laws, anyone? Let's start with the first law of dominance. What is law of dominance? What does the word says?
About the
One
dominant trait.
Sorry, I could not hear Reem completely. I don't know if it was my network or hers.
One trait over the another one, like it's dominant over the other one.
Yes, trait. So you all are, so to explain this, let me first confirm that you all understand these 3 things or not. Do you all understand what is a gene?
Yes.
What is a gene? Quickly, Reem. Tell me what it is. What do you understand what it is? Is it something physical or is it just an idea?
Gene are the traits that we get from our parents.
Yes, gene codes for those traits. You are correct. But what are genes? Like if I have to find a gene, let's say if I have to touch a gene, can I do that? Is it somewhere present in the cell?
Yes, I guess.
Madhya says no, right? Madhya, you says no?
Yes, you can't touch the gene. The gene is present in the nucleus.
So it is just an
It's
idea.
present in the cell.
It is, if so, is it something physical or is it just an thing that we are assuming?
It is physical.
It is physical.
So if it is physical, we can touch it, no? Like not like I can touch it with my finger, but it is there. Gene is nothing but a part, a segment of DNA itself. Can't we touch the DNA? Can't we have DNA in our hand or in a test tube? Yes, we can have it, right? We cannot see it with our naked eye because it's very, very microscopic, tiny. It's a molecule. So we cannot see molecules. When we are touching water, actually we are touching the molecules of the water, right? But we see it as one liquid. We do not see the molecules. Gene is just DNA, where it has a starting point and an end point, which is called the start of the gene and the end of the gene. And in between, everything codes for a functional protein. That's called a gene. So DNA is a very long thread. Everyone understands that? Let me use this wire to explain DNA to you. Can you all see this cord, this wire? I think is it visible to you, this white cord?
Yes, sir.
Yes.
Yeah. Let's say this segment that I have looped out here. Can you see the loop?
Yes.
So this loop is a gene. It starts from here and ends here. Before that and after that, the part of DNA does not code for anything, like does not code for a protein. So that will not be called a gene, but it is a DNA. This whole thread is a DNA molecule, a very long molecule, polymer. And a part of it which codes for a protein is called a gene. So it is a physical thing. Is it clear, Madhya? We can touch it. Do you understand? The concepts are clear?
Yes.
So DNA gets transferred in the form of chromosomes from gametes, like from mother and father to the embryo. Yes, you all understand that also?
Yes, sir.
Yes.
And what are chromosomes? So gene is clear. I'm happy. Everyone knows gene. Everyone knows chromosome. What is a chromosome?
Chromosomes are thread-like structures that have genes and they're responsible for transfer of characteristics.
Yes. So which means if again, this is the DNA. Okay. Now it is very long, but the cell is not very long to keep this thread as it is. So what can we do? Have you ever seen a spool of wool or a thread? Everyone, people?
Yes.
Yes, sir.
Yes.
So there is something like a cylindrical thing, right? And then on top of it, we just keep wrapping, coiling and coiling and coiling a long thread, right? This way, we can keep a very long thread, maybe a kilometer long thread in just 2 centimeter space. Yes or no?
Yes, sir.
Yeah. So this is exactly what is chromosome. So this is the DNA. It cannot be kept like this in the cell because it occupies lots of space. So we are going to supercoil it like this. So when we keep on supercoiling and just imagine this, so this is how it forms, right? And just imagine a long thread like this. You understand? Everyone? A supercoiled
Yes.
structure of DNA is called chromosome. Is it clear? It looks like a thread because it is very thick, like its downside like this. Do you understand, everyone?
Yes, sir.
Yeah. So chromosome is nothing but a supercoiled DNA. But to coil it, we need something, right? Just like to coil this thread, we need a base on which it can be coiled, right? DNA cannot be coiled in the air on itself. So it also needs something on which it can be coiled, right? Yes or no?
Yes.
What can we coil it on? Proteins, right? There are DNA binding proteins called histone proteins on which you can bind a DNA and keep it in a very less space in the cell. That is called chromosome. So chromosome is DNA plus protein. Everyone is clear?
Yes, sir.
Yes, sir.
So whenever we have to send something to another cell or to a gamete or through gamete to a zygote, we send it in the form of a packed. When you send a gift to someone, you pack it, right? You make it short, pack it somewhere, make a covering and send it to. That person will again open it and see what is the information inside, right?
Yes, sir.
The same thing cells are doing here. So gene is clear, chromosome is clear, right? Let me also ask you, do you understand alleles? All these are concepts from previous class. I'm just confirming. If you don't know it, I'll explain it. If you know it, then I'll go with the laws. Do you know what are alleles, everyone?
So genes and alleles are same, right?
Are they same? Then why do we have two different words? You are correct. But there is also a twist to it. You're correct that genes and alleles are same. But then why do we have alleles? What are alleles? Anyone knows the difference between the two, the genes and alleles? Come here. Look at this character. You see this? Character is for plant height. Yes?
Yes.
Now, if there is a gene which tells what will be the height of the plant, will it be a single, just one gene? Because if it is one gene, either it can be tall or dwarf. How both are present? Both are possible. How can some plants be tall and some plants be short? Does it click? Anyone coming to an answer? What are alleles?
Because there are two
Alleles are only
traits.
one pair of characters.
Yes, you both are close, but you're not, you're beating around the bush. Let me tell you, alleles are just two different or two or multiple different forms of the same gene. Okay. Is it clear? For example,
Yes, sir.
you have the same cap, but in 2 or 3 different colors. So the cap is exactly same. The only difference is one is blue Adidas, the other is black Adidas, one is beige Adidas. You understand?
Yes.
Similarly, a allele is just the gene of height. So if I'm talking about alleles of the character of height, then I have two alleles. One allele is capital T. Okay, whatever gene which gives rise to capital T, and the another allele is small t. You understand? Basically, both these genes are the same gene of height, but they have some differences in between, where one gives rise to tall character and the other gives rise to short character, but both are the height, both are the gene for height. Correct? You understand?
Yes, sir.
Yes.
So write down alleles. If you don't write down, alleles are alternate versions. Alleles are alternate versions of the same gene. Which gives rise to contrasting characters. Contrasting characters. Is it clear, everyone?
Yes.
Perfect. Now let's come to the laws, because we'll be using gene, allele, chromosome, all this a lot while discussing the laws. Okay. So the first law of dominance. Okay, come back, Reem. What were you saying about the law of dominance? You were telling
That
something.
one trait takes over the another, like it's dominant over the another one.
Yes, perfect. Which means, this means that there are two alleles, right? Now we will talk in terms of allele and you all will understand, right? If there is any confusion, please ask.
Yes.
So law of dominance says that for every gene, for every gene which has more than one allele, which means more than one version, then always one version is dominant over the other. For example, if there is a capital T given by the mother, like the female plant, which is crossed with a small t given by the male plant, then all the babies will have this as a diploid, right? In gametes, it's haploid, but it will be this in the first generation, right? F1.
Yes.
Always, whenever two alleles are present, always one allele, which is this one, it will be dominant. It will show its effect, and the other will be masked, which will be called recessive. Is it clear, everyone? This was law of dominance, that when there are more than one versions of the gene, which are called alleles, then one allele dominates the expression of the other. Okay. So how will the recessive allele ever be expressed? Do you know that? Now for that,
In next generation.
Yes. Why in next generation? So it's a very right answer in next generation. But what happens in next generation that it gets expressed? Can't it get expressed in the first generation only? The way we understand concepts are slightly flawed because it makes it easier. But I will explain you things where you can, from any generation, you will understand what's happening actually. Yes, Reem, first complete your answer, then I'll come to it.
In first generation, the another one is the dominant. So in next generation, the recessive will be the dominant one.
Yes, but this definition comes with a very fine concept. Let me tell you that. There are two types of dominant traits. Okay, one is called pure line trait. A pure line trait is this, capital T and capital T, right? You understand why this is a pure line?
Yes, sir.
Why?
Because it's dominant.
No, because both are same. The mother is also giving this trait allele and the father is also giving this allele to this particular individual. That's why it's called a pure line trait. So it will be a homozygous dominant. Do you understand the term homozygous dominant?
Yeah, because it's same.
Yes, see, in genetics, and Madhya, pay attention to it. In genetics, it's all about the concept. Once you get the concept, whatever question they ask you, you are going to solve it. No problem at all. Genetics is all about the foundation. The building is not taught by teachers. It cannot be taught by anyone because you can ask thousands of different questions on the same foundation. It's just like the rule. I think genetics is the only very, very mathematical and conceptual part of biology where you don't have to mug up. That's why it's very interesting. But Madhya, you know, you found it a little difficult, right? So I'm keeping this in mind and starting from the scratch. Are you following, Madhya? Are you there?
Yes, sir.
If you are fluent in genetics, you know, I'll be very happy. And I think everyone will understand. So you are my benchmark. If I manage to make you understand, it's done. Okay, Madhya? So you are with me?
Okay, sir.
So this is called a pure line trait because it's homozygous dominant. Okay. There can be another pure line trait. Can you tell me what it can be, Madhya? If capital T is a pure line.
Small t, small t.
Yes, perfect. And what will this be? A
Homozygous recessive.
Yes, correct. Now let me tell you one simple thing that you have to remember. If a trait is recessive, it can only be expressed when it is present in a pure line or a homozygous state. Is it clear, everyone? Write down. A recessive trait can only be expressed in a pure line or a homozygous condition. Make sense to you, Madhya and Reem and others?
Yes.
Perfect. And then there is a hybrid trait. What do I mean by hybrid trait? What will be the genotype of a hybrid trait?
Capital T and small t.
Perfect. Now, this is heterozygous, right?
Yes.
But now Madhya will tell me, will it be heterozygous dominant or heterozygous recessive?
Heterozygous dominant.
Perfect. There is nothing like a heterozygous recessive. A recessive condition cannot express in a heterozygous condition. You understand, Madhya?
Yes.
This exact thing, the hybrid trait where it is heterozygous but still dominant, is the basis of law of dominance. Because here, this is expressing and this is not. Make sense?
Yes.
Everyone?
Yes.
Clear? Perfect. Now let me give you an exercise. Tell me, the question is, everyone has to do it, and I'll ask for this one, I'll ask the answer from, I want the answer from Minashi. Everyone has to, Memuna, welcome, how are you? You've just joined, yeah? Who has joined just now, Minashi or Memuna? I think it's Memuna.
No, sir, I'm there from start.
Okay, you're there, okay? Okay, good. So the question is, I have two plants, okay? One plant is, the father is hybrid tall. Okay. Father means the male plant. And the female plant is also hybrid tall. Both are tall, okay? But I'm giving you hybrid tall. Now tell me, when they will be crossed in generation first, F1 generation, how many plants will be tall and how many plants will be short? I want the answer from Minashi. Minashi, can you speak? Is your mic working? Yes, perfect. Please do it fast and tell me the answer.
3 tall plants and 1 short plant.
Sorry?
3 tall plants and 1 short plant.
3 tall plants and 1 is short plants. I want the confirmation from Madhya. Madhya, is she correct? Will there be a short plant in generation? But I think Reem said that in first generation, no recessive trait is visible. Reem, would you like to pitch in the discussion and tell me?
I guess it's correct, sir.
You guess it's correct? If these are the parents, then in first generation, you are saying that there will be a short plant also?
Yeah.
But you said that there is no recessive gene which can be expressed in the first generation.
I guess I'm wrong.
No, you are correct. You're not wrong.
Oh,
The way
okay.
these concepts are taught is slightly flawed, I told you, right? We always in the books start from a pure line trait, always. That's why, let me go. So you are right. In F1, there will be 3 tall and 1 short. If this is the parent given. So always in your entrance exams and in your boards, pay attention to what are the parents' genotype. Is it clear, everyone? Now let's solve this first. Then I'll go to how Reem is correct. Okay. So if the father is hybrid tall, what will be the genotype? Madhya?
Capital T, small t.
Okay, perfect. And what are we crossing it with?
Small t and capital T.
Yeah, we always write dominant first. Okay. So let's say the same thing, right? You're saying the same thing?
Okay. Yes.
Yeah. So which means this is the male parent. I always use the word father and mother for plants. Then I think it's not good. Male parent and the female parent plant. So we are crossing it. Okay. Now when we cross it, we always cross it through Punnett Square, right? You know what is a Punnett Square, everyone? In your junior classes, you have done it. So basically, when we make a Punnett Square, what we are doing is we are making the gametes. So on this side is the gametes that can be made by the male, and on this side is the gametes that can be made by the female. Now remember, if this is a diploid cell, if both the alleles are present together, of course they are present in the body, not in the gamete, because in gamete only 1 copy is present. Yes or no? Chapter 3.
Yes.
Yes. So what all gametes can this father make? Either in one gamete it can send capital T, in another gamete it can send small t. Yes?
Yes, sir.
The same thing the female can also do. These are the all possible gametes, right?
Yes.
Now let's start making the progenies. So it will be capital T, capital T,
Yes.
capital T, small t. Here also capital T, small t. And in this case, it will be small t and small t, right? So if this is the parent generation, then after crossing them both, in first generation, which is known as F1, we get a small plant. Now, there are 2 terms associated. Okay, pay attention. One is called genotype, and one is called phenotype. And these ratios are always asked in boards and in entrances. So how to figure out? Genotype is just about the genes. It has nothing to do with what it gives rise to, a tall or a short or whatever. So how many different kinds of genotypes can you see in this 4 progenies? One is capital T, right? Yes.
1, 2, and 1.
Yes, right. So 1 is to 2, you mean to say, is to 1, right?
Yes.
This is the genotypic ratio. Do you understand, Madhya?
Yes.
This is for 1. What is 1? Capital T is 1, is 2. Capital T, small t are 2, right? 2 offsprings will have this genotype. And then only 1 offspring will have this genotype, right? Is the ratio clear? Now,
Yes.
what is phenotype? Phenotype is what are the visible characters, the pheno. The word phenology is the study of visible characters. Phenology. Pheno means visible characters. Logy means study. Biology, study of bio. Bio means life. Cytology, study of cell. Anything. You understood? Phenotype. Can anyone tell me what the phenotype ratio will be? What are the 2 possible phenotypes? Either it will be tall.
3 and 1.
Yes. or it will be short, right?
Yeah.
So how many tall plants can I see? I can see this one as tall because it is pure line tall. I can also see these two as tall. But what is the difference between these two and this one, Madhya? All three are tall, but what is the difference?
Their genotype are different.
Yes, their genotypes are different. Madhya, can you see? In this case, it is a pure
Yes.
line. The thing that we were talking about here, a pure line trait, right? Homozygous dominant, pure line tall. But in these two cases, it is a hybrid tall, where the law of dominance is playing its role. Make sense, everyone?
Yes.
Yes.
Yes. And this one is a homozygous recessive, a pure line short. So what will be the phenotypic ratio? 3:1. Is these concepts clear to you?
Yes, sir.
Yes.
Now let me come back to what books tell and how Ream is not incorrect. So whenever we start the study, we always, if we are talking about two genes, then we have to start from the pure line characters. Otherwise, that study will not go, like it will be confusing. Like this is where I'm not starting from pure line parents. The parents themselves are hybrid. And this is an actual question that has come in boards, and lot of students just see that, okay, parents and F1 progeny, oh, all will be tall and 0 marks. But you have to see the parents are not pure to begin with. Books always starts from the beginning. This is how they teach us, right? Education is one step at a time. Yes or no?
Yes.
But entrance is not about one step at a time. They can ask you anything from anywhere. So now let's start what book says. So if I want to make a cross between a tall and a short plant. So what is this? This is a pure line tall, right?
Pure line.
Pure line tall or homozygous tall. And what is this? A pure line short or a homozygous short. Now, if I have one parent tall and one parent short, it is very obvious to expect that some of the kids will be short and some of the kids will be tall, right? If you do not know genetics, then if I ask you that, see, one parent has blue eyes and the other parent had black eyes, and they have, let's say, 6 kids. So what is the chances of blue and black eyes? So even if you don't know, you will just logically say that, some will have blue eyes and some will have black eyes, right? Yes?
Yes.
Yes, sir.
But the important information missing here is whether the parents are pure line for their trait or not. So this is where all the book starts. So they always consider parent generation to be a pure line tall. So write down. If there is a homozygous tall and a homozygous short. Now let's see why Reem is not incorrect and she's correct. Because once you will do this, I will, for the first few Punnett squares, I will go right from the scratch. Okay. After that, when you will understand, I will just assume things and you will just be fluent with it. This parent, the male, let's say, the male parent, what different gametes can it form? Either
Capital T and capital T.
Yes. There is only one type of gamete possible, right? Because it's pure line. What type of gamete this can form? Small t and small t, right? Yes.
Yes.
All the progenies will be capital T small t, capital T small t, capital T small t, and capital T small t, right? And this is generation 1. You know why we use F? F stands for, anyone knows? F stands for filial. Filial means generation. So F1 means filial 1, which means generation 1. Okay, everyone? So in F1, tell me what will be the genotype? Madhya. what will be the genotypic ratio, Madhya?
1:1:1:1.
No, there is no point of putting is to now. They're all same.
Yes.
The genotype, there will be no genotypic ratio actually, or it will be just one. Yes, all are same
Yes.
genotypically. And if genotypically things are same, then it is a rule of thumb that phenotypically they have to be same. But the opposite is not true. Make sense, Madhya?
Yes.
So if all genotypically are capital T small t, then all phenotypically will be tall. Yes? Yes or no?
Yes.
But what kind of tall they are? Are they like their parent tall? What is the difference? Are all these four offsprings tall, but are they like the parent? Are they like the father?
They are heterozygous.
Exactly. The phenotype is same, but the genotype is actually different. So you see, all the offsprings are tall, but all of them are hybrid tall. Whereas their father was a pure line tall. Why offsprings got hybrid tall? Because the father, which was a pure line tall, got crossed with a mother, like the female, which was pure line short. So all of them were hybrid, and because we know that capital T is dominant over small t, in all the cases, no recessive trait will be visible. So in F1, all will be tall and no one will be short. Do you understand, Dream, why you are not incorrect?
Yes, I do.
Yes, so you are correct that in F1, no recessive trait is shown. But remember, for that, the parents have to be pure line. If they ask you with this parent combination, in the next generation, you will get short. Is it clear, everyone?
Yes, sir.
So books, what they do is, as the second step to get F2, they self-cross F1. This is how they explain you the concept. So when you self-cross F1, what are you doing basically? You are doing this cross. Am I correct?
Yes, sir.
And is this exactly the same that we did already above? Isn't this same like this? This is the same thing, right?
Yes, sir.
Yes.
But here we considered this generation as the parent.
Pure line.
No, we considered this generation as the parent, not pure line. If we consider pure line as the parent, then this cross will be a self cross of F1. And then as a result of this cross, what we will get? We will get 3:1 ratio of genotype. Make sense, everyone? Madhiha, is it clear?
Yes,
Sorry,
sir.
I'm sorry. I made a mistake here. 1:2:1 ratio of genotype, correct?
Yes.
And what will be the phenotype? 3:1 of the phenotype. So for the first time, this one, which is a recessive, will be shown in F2. So that's why we say in books that recessive characters are not shown till generation 2. And we remember this line. But what we forget to remember and understand is that this makes sense only when we start with pure line parents, both tall and short. Okay? So calling someone parent generation or F1 generation or F2 generation is very subjective. Entrances can tell you, they can give you any combination. Is everything clear to everyone? Madhya?
Yes.
Yes.
Now let me test your concepts. Let me give you a question. Write down. A hybrid tall pea plant, a hybrid tall pea plant is crossed with a pure line short pea plant. Is crossed with pure line short pea plant. Also write the another version of the question. A heterozygous tall is crossed with a homozygous short. Okay, do you understand? Both are the same things? Yes or no?
Yes, yes.
Yes. Tell me, if this is the parent generation, I'm considering this as a parent. If this is parent generation, what will be the genotypic and phenotypic ratio in F1? You have to find GR and PR in F1. And I would like if the answer comes from Madiha. Others can write it in the chat, okay? Please, just to me, not to everyone. Just write your answers in the chat. I just want Madiha to answer it. Madiha, if you do not feel answering like in vocally, you can also write in the chat. But I'll wait till your answer comes.
Okay, just 2 minutes.
Yes. Write the see, there is one rule. Write the genotype of the parents, make a Punnett square where you will segregate the gametes, then make a cross and find the answer. This is a slow mode, but first do that. Over the time, by the time this chapter ends, you will be fluent. You can do that in your mind, and I will train you for that. I'm waiting.
Should we find F2 also?
Just the F1. How will you find F2? I have not told you whether F2 will have a self cross or something else, right? We just by default imagine that F2 is the self cross of F1, right? Yes or no?
Yes, sir.
Saifullah. So in my classes, you also have to understand what is in NCERT, how is this concept like presented to you, and what are all the advanced versions of it which can be asked in entrances. You understand? So mostly when they talk about F2, it is the self cross of F1, given rule. But right now find F1. I want everyone's answer in the chat. I've opened it. By the way, when I open this chat window, can you see this chat window? No, no.
No.
Can you see something on the screen? Okay, good. Otherwise, it does not make sense, no? Yeah. Yes. I received Madiha's answer for GR. What about FR? Yeah, sorry, PR. Madiha. I received Saifullah's and Minashi's answer. Reem, I'm waiting for you. It's better to take time than to be wrong initially. Once you understand the concepts, you can be just faster and faster. It's just practice after that. Right now, it's concept. Are you solving it, Reem?
Yes, sir.
Yes, yes, please. I will allow you sweet time in the beginning. Do not worry. Are you finding any difficulty, Reem?
Kind of.
Okay. So would you like if you do it with me?
Yes.
Okay, let's do it together. So let's first decode what this sentence, the word problem, means in terms of genotypes. So talking about this, let's say this is the male one. And you can just assume anyone as, you can also say A or B, parent A, B. You know, I just like to use these symbols. So what will be the genotype of the male here? Quickly, Madhya. Okay, Reem. Okay, you unmuted yourself first. Tell me, if it's a hybrid.
Capital T
Yes.
and small t.
Perfect. And what will be for this?
Small t and small t.
Yeah, then what is the issue then? You did it. You solved it already. If you understand this, it's done. Now let's see how it's done. Tell me, what will be the possible gametes for the male? Reem?
Capital T, small t.
Perfect. And for the female?
Small t and small t.
Perfect. Then what is the problem? First kid could be capital T small t. The other can be capital T small t. This will be small t and small t small t. Any problem?
No.
What will be the GR? These two are same.
2.
And these two are
2.
same.
So 2 and 2.
2, 1, 2. How 2, 1, 2? It cannot be 2, 1, 2, no? These two are same and these two are same. Right?
Yes. So
It's 2 is
1.
to, so this is to this ratio we have to find. There are only 4 offsprings. And if 2 are of one type and 2 are of other type, 4 are done. Understood, Reem?
Yes.
What about the phenotypic ratio? Now, phenotype can either be tall or short. So how many of them are tall?
2.
Which one? These two?
Yes.
Or these two?
No,
These
no.
two.
Above.
Yeah. And what kind of tall they are?
Hybrid.
Yes. And how many are short?
2.
And what kind of short they are?
Pure line.
There's only one kind of short. Okay.
Yes.
So if now another, write down a neat question based on this problem, a neat type question. Okay. Advanced level question. So the question will not be this easy. The question will be, if a hybrid tall is crossed with a pure line short, the tall of the F1 generation will be genotypically same as the parent, different from the parent, both, none. So these two tall things here, these are the two tall plants, right? Two tall offsprings. Can you all see this, Madhiha?
Yes.
These two tall, are they same like the parent or different from the parent genotypically?
Yes.
Same. But if the same question was asked for this cross, were the tall
They're not same.
like the parent?
No.
No. Here it was hybrid tall and the parent was pure line tall genotypically. But in this case, the tall 2:2 ratio, the tall are all hybrid tall, right? Yes or no, everyone?
Yes.
And the father or the parent was also hybrid tall. Yes?
Yes.
Everyone understood the concept that whenever we talk about F1, we have to first get to know what are the parents. Do not blindly remember lines about F1 or F2. Yes?
Yes.
And this is a better way to remember. Very good. So in this case, if these are the parents, then in F1, what is the ratio? 2:2. We ourselves figured out 3 different scenarios. Let's start from the beginning. If the parents are both hybrid to begin with, then the ratio will be 1:1, 1:2:1 for genotype and 3:1 for phenotype in the F1. Yes?
Yes.
If the parents to begin with are pure line, both of them, then it will be 1 and 1, no ratio at all. The ratio will only come in F2. Yes? And if the third scenario, if the question comes like this, if one is hybrid and one is pure line, then we will get this ratio. Yes? Are you finding it interesting and is it easy to understand? No mugging up, no cramming, just concepts. Madhya, how are you finding it?
I am interested.
It's easy?
Yes.
Okay, now for Reem, because Reem struggled a little bit in this one. So I'll give you one last problem. Okay, everyone, would you like to do it?
Yes, sir.
Yes. Now write down another question. If a pure line tall, plant is crossed with a heterozygous plant. If a pure line tall plant is crossed with a heterozygous plant, so considering this as the parent generation, what will be the GR and PR in the next generation? Everyone understood the question?
Yes.
Yes. So it is pure line tall crossed with heterozygous. Please do and tell me, considering this as the parent generation, tell me what will be the GR and PR in F1. Again, please give your answers in the chat. Okay, I'm getting some interesting answers. Minashi and Reem. Mamuna, you're there? Mamuna, you're not speaking.
Yes, sir.
And I realized you are also not answering in the chat. Why? Any difficulty you are finding?
No, no.
No, everything is clear, right?
Yes, sir.
Very good. Minashi? Okay, so let's go and solve it again. Okay, I got all sorts of answers, like all answers possible. Some or the other student has given everything. Now everything cannot be correct in biology. Okay, in logic, let's go to see what is correct. Again, you all are not following the procedure. Now repeat with me. First step, decode the word into alleles. Write it down. Decode the words into alleles. First step. Everyone has written. Now I know that I'm doing some primary class thing, but it works. For any higher concept, we have to start like this. I was taught in my MSc research concepts like this. First thing first, decode the word expression into allele, genotype. Second, make a Punnett square, write the gametes possible, and do the cross. Written, everyone? Now, I know that for someone it must be very easy, for someone it must be a little confusing, but we all have to be on the same page before I go to other laws. And even convoluted than this, complex than this, is recombination, mutation, and other things, linkage. So let's first be on the same page. Okay, I would like Minashi now to follow with me. Minashi, are you there?
Yes,
Let's solve it together. Okay, you and me. Okay. Now tell me, if I consider this as parent A and this as parent B this time for a change, what will be the genotype for parent A?
capital T, capital T.
Okay. And for B?
Capital T and small t.
Then where is the problem? I don't understand. All of you are doing this correct. Then how can you manage to find the answer wrong? You know, I think you're not paying enough attention. Now look here. Is this correct, everyone? Is Minashi
Yes, sir.
correct? Yes. Now, if we are crossing them together, then let's make the Punnett square. What could be the possible alleles or the gametes for A? Minashi?
Capital T and capital T.
Perfect. And on this side for B?
Capital T and small t.
Okay. Then what will be the first progeny? Cross this and this.
Capital T and capital T.
Also tell me whether it will be pure line tall, pure line short, hybrid tall.
Pure line tall.
Okay. And what about this second progeny? What is the
Capital
genotype?
T and small t.
What about this? What will be
Heterozygous.
tall? Yes?
Yes.
Perfect. About this one?
Capital T and capital T. Pure line tall.
Yes. And this one?
Capital T and small t. Heterozygous tall.
Okay. So first talking about the GR, the genotypic ratio. How many different types of genotype do you see? I see this one, capital T is one possibility. What is the other possibility?
Capital T and small t.
Okay. What is the other possibility?
That much.
No other possibility, right?
Yes.
And how many are capital T out of 4?
2.
How many are capital T small t out of 4?
2.
So what will be the genotypic ratio?
2:2.
And do you understand why is it 2:2?
Yes.
Did you understand this, Reem, why it is 2:2?
Yes, sir.
Yes?
Yes.
Perfectly clear?
Yes.
Very good. I know that Reem is smiling right now. See, I knew that. I'm good at reading minds. I can see, you through the screen. Let's come to PR. Now Reem will answer. What could be the possible phenotypes? Either tall or short, right?
Yes.
How many
So
do you think are
2:2.
tall?
2:2.
So you think 2 are tall and 2 are short?
Yes.
Phenotype is 1.
Madhya, you understood, Madhya? Reem, look here. All are tall. Can you see?
Yes. Yes.
These 2 are pure line tall. Look here. These 2 are pure line tall. Yes?
Yes.
And following the law of dominance, these 2 are
Hetero.
hybrid tall. Right?
Yes.
But all are tall. So what will be the phenotypic ratio between tall and short?
1:1.
1:0. Why 1:1? 1 means there is one short also. And 1:0 is simply 1, no?
Yeah.
Is it making sense to you, Reem?
Yes.
Now, if I ask the same question that was asked in entrance, that the same word problem, if a pure line tall is crossed with a heterozygous plant, then the tall plants of the next generation will be same as the parent, different from the parent, both, None.
They
What
will
do you
be both.
Yes. Because what was the parent tall like?
They
Capital
were
T and
pure
capital
line
T.
and they were heterozygous both.
Yes. This was also tall, right? But pure line tall. And this was also tall, but it was hybrid tall. Makes sense, everyone? And in F1 also we got a pure line tall and a hybrid tall, which is same like the parents. Yes, you understand? So we also got things which are same like this parent and also which are same like this parent. Make sense to you, all of you?
Yes, sir.
Reem. Now please look at all the four problems that you just solved, because I'm going to make it a little complex now by bringing in there's something called exceptions to the law. Biology is a science of exceptions. There are certain things that do not follow the rule. Okay, so there are certain laws, there are certain phenomena in biology that do not follow. So law of dominance, we are still on law of dominance. I think we have discussed 4 possible problems which are actual questions from the boards, and all of you understand this, right?
Yes.
Yes, sir.
Yes.
Now write down something about law of dominance. Write down. Law of dominance states that one allele is dominant and always will express itself. One allele is dominant and always will express itself when present in a heterozygous condition. When present in a heterozygous condition with another allele. And in brackets write down recessive allele. So the other one will be recessive allele. Okay. Written, everyone?
Sir, can you repeat the sentence once?
Yes, that is a little difficult. Law of dominant states that, what did I say? Did someone write it? I just want it to be exactly same. So if I'll speak it 3 times, it will be chances are it will be difficult. Reem, can you repeat? Have you written, Reem?
Yes, sir.
Yes. Can you please repeat
Law
from
of dominance states that the one allele expresses
itself
always.
I think expresses itself always. You can't read your own handwriting. What?
Express itself when present in a heterozygous condition.
Yes.
Then in bracket, with another allele, then in bracket recessive allele.
Madhya, done?
Yes.
Yes. Write down another line. According to this law, no, not according to, as a result of this law, a recessive trait can only express when present in a homozygous condition. In brackets, you can write down small t, just for your reference. Is it okay, everyone? Do you all understand this?
Yes, sir.
Shall I move forward?
Yes, sir.
Now here comes the moment you think you have understood something. In this chapter, I will throw something at you which will make it difficult. So write down exceptions of law of dominance. No, before that, write down law of dominance is not a pure law. It has exceptions. Okay. When I say something is a pure law, or sorry, universal, the word, another word for pure in books is universal law. So write down law of dominance is not a universal law. It has exceptions. Just okay. Now write down exceptions. And a lot of question comes on exceptions. Exceptions of law of dominance. Now it's very simple. If I say there is a law which is to follow my command, what could be the exception of that law? If there is a law that everyone has to follow my command, I am the most dominant one, you all have to follow my command, and you all are doing it. So you all are following the law. What could be the exception of that law, logically? Simple. Saifullah, what can you do to prove yourself an exception to the law of following my command?
By not listening to you.
By not following the command. Perfect.
Yes.
Or anything that you can do to become an exception? To follow it, but not properly, right?
Yeah.
Yes or no? Yeah. Like I said, go to the market and buy me mangoes. So Reem either just said, okay, I will not go to the market and not buy mangoes for you. That's complete, just you're saying no to that command. Or you can say, I will go to the market, but not bring you mangoes. You understand? So the first one is called, write down, exception of law of dominance is, first is incomplete dominance. Incomplete dominance. Now, what do we mean by incomplete dominance? What does the word says? The word still have dominance in it, right? So there is dominance, see, but it is incomplete. What does it mean? Write down. It means blending of traits. Okay. Blending of traits. Is it clear, everyone? Am I audible?
Hello.
Yes. Am I audible?
Yes, sir.
Yes, sir.
Was I audible throughout, or did you lose me?
My connection cut off, so I was not able to.
Okay. For Reem and Madhya, was I audible throughout?
Yes.
Yes. So incomplete dominance means blending of traits. Now, let's take a situation here. If, like, this is a situation we are taking, let's assume. So capital T, if present with capital T, is pure line dominant. Yes. Madhya, pay attention to this point. If capital T is present with capital T, it is pure line dominant, right? No question.
Yes.
If capital T is present with small t, it will be hybrid tall. Still the law of dominance is being followed, right?
Yes.
But if there is a situation where, let's take an example of a flower color. So it happens. So let me tell you, there is a flower for which there are two colors. The allele R stands for red, and this is dominant. Okay, I know that this is dominant allele. Okay, everyone.
Yes.
Then small r will be for white. Okay, you understand?
Yes, sir.
Which is recessive, but we denote the same letter for the sake of, like, there should be no confusion. If capital R is for red, then we will not use W for white, or we can do the otherwise. Actually, this is not the correct notation. This is given in NCERT, but internationally in the textbooks, if you'll go and study higher genetics, if anyone from you wants to go into research and study higher genetics, remember that the traits are always named, the letters are always named with respect to the recessive trait. So all of this that we studied from NCERT, we give T to tall, right? Actually, it's capital D for tall. You know why? Because small d is for dwarf. This is the correct notation in genetics. You understand, everyone?
Yes, sir.
I'm just telling you. So NCERT is outdated in this context. You know, I don't know why they have not changed it. Have they changed it now? Can someone confirm? Because I think they should have, or but tall is capital D, dwarf is small d. Similarly, if I want to write the right notation for red and white, what should be the red? Capital W for red, right? You understand?
Yes, sir.
And small w for white. So this is but this is easier to understand. That's why we follow. The main thing is you should understand the concept. Okay. So let's stick to this one, the NCERT one. R is for red, dominant. So in this case, what does law of incomplete dominance says is that if there is this combination, then of course it is going to be homozygous red, or we can call it pure line red. Everyone understands? Everyone, is it
Yes,
clear?
sir.
And if this is this combination, what is this going to be? Homozygous recessive or pure line recessive. Okay. But the catch is in this condition. Normally, following the rules of law of dominance, what should be this genotype giving rise to? Madhya, what should be the phenotype of this genotype according to law of dominance?
Hybrid.
Hybrid what? Red, white?
Hybrid red.
Hybrid red, right? It should still be red. The flower should still be red in color, just hybrid in genotype, right?
Yes.
But here, the flower is pink in color. You know what has happened? So this gives rise to pink colored flowers. Here it is incomplete dominance. The capital R is not able to show its dominancy over small r when they are present together. So when they are present together, the small r, the recessive allele, is saying, enough of your dominance, of your bullying. I will not submit to your bullying, and I am not following the law of dominance. Okay, you cannot dominate over me. So let's be together. You understand that?
Yes.
So the only way to, like, the dominant one is dominant, like it's arrogant. It's like, no, I will provide. So what is happening on a cellular level is this R gene, the capital R gene, it must be providing some pigments, right? Producing some proteins or pigments which give red color to a flower. Yes? Yes or no? And the small r must be producing some protein which is giving white color to a flower. White color does not mean no color. White is a color. There are pigment for white, right? No color is transparent. Is it clear, everyone? Are you understanding, Madiha?
Yes, sir.
Yes.
When both of them are present together, the white, even the recessive one is producing some pigments, and the red one is producing some pigments. These pigments are blending together to give a pink color to the flower. So in this case, can you see the dominancy? No, the dominancy is blending. So this example is an exception to the law of dominance. Okay. Is it clear?
Yes, sir.
Now write down. In incomplete dominance, the dominant trait is unable to show complete dominancy. The dominant trait is unable to show complete dominancy over the recessive allele. Sorry, change the word trait with allele. The dominant allele is unable to show complete dominancy over the recessive allele when both are present in a heterozygous condition. When both are present in a heterozygous condition. Next line, as a result, blending of traits takes place. As a result, blending of traits takes place. Is it clear?
Yes, sir.
Now this was giving an example of the color. Now it's easy to understand how color blends. When white and red blends, what does it makes? Pink. You all have painted, right, as a kid? If you do not have a pink crayon, just rub white over the red one and it will become pink smudge, right? So we know it's blending. Now, write a question and give me the answer if you have understood. Write down the question. Shall I dictate the question, Madiha, Minashi? Write down. A pure line tall, or let's write down in biological terms now, a homozygous tall plant is crossed with a homozygous short plant. A homozygous tall plant is crossed with a homozygous short plant. Given that there is incomplete dominance in this species, given that there is incomplete dominance in this species, find out the genotypic ratio and the phenotypic ratio in the next generation. Okay, so consider this as the parent generation. Then find out the genotypic ratio and the phenotypic ratio in the next generation. Question is clear to all of you? Just say yes or no. Madiha, is it clear to you?
Yes.
Now please find out and give me the answer in the chat. Now you have to factor in the blending. Okay, everyone. Just by including one sentence, genotype, the incomplete dominance, they have changed everything. Okay, I'll wait for everyone's answer to come, then we will do this. Sefula, you understood the concept?
Yes, sir.
If you pay attention to these three laws, then understanding recombination and linkage and everything ahead will become easier. Others finding any problem? Madiha? Kind of? Like, are you stuck somewhere? Then you will solve with me. Are you stuck somewhere, Madhya?
Yeah.
Okay. And I think Reem just cropped out. Mamuna, what about you? Okay, I received your answer. So let's do it. Okay, Madhya, you will help me, okay? Again, same concept we will follow. First thing, let's say this is parent A and this is parent B. Then tell me the genotype for parent A. Madhya?
Capital T, capital T.
Capital T, capital T. And for parent B?
Small t, small t.
Small t, small t. And we are crossing them, right? So let's come to the Punnett square part. So till the Punnett square thing, everything is going to be the same, no problem. Okay. So what are the possible gametes that parent A can make? Madhya?
Capital T, small t.
How can parent A make small t?
Sorry, capital T, capital T.
Yes. And so we will, on this side, we will write of A, and on this side, we will write of B. So about B, tell me. Madhya?
Small t, small t.
What will be the first? Continue, keep saying.
Capital T, capital T.
Capital T, small t, this and this.
Oh, capital T, small t.
And then for the second?
Same, capital T, small t.
Yes. And for the third, this and this?
That is also capital T, small t.
And for fourth?
Capital T, small t.
Yes. So clearly, what is the genotypic ratio?
1.
Perfect. And what is the phenotypic ratio?
That is also 1.
That is also 1. But tell me what, so this was a pure line tall, right? And this was a pure line short. And you are saying that here the genotype is capital T and small t, but the phenotypic ratio is also 1. But what is this one?
They're all tall, heterozygous tall.
No, they're all medium height. You did not factor in incomplete dominance, right?
Yes.
I told you that there is an incomplete dominance in the species, but in the first generation, if we start with pure line tall and pure line short, nothing makes much of a difference. It's just like a normal cross. So everyone forgets the point of incomplete dominance, but all will be medium plant. No one will be pure line tall, no one will be pure line short, because it will be incomplete dominance. Now, I'm just complicating it. If F1 is self-crossed, then now tell me in F2 what will be the genotypic ratio and phenotypic ratio? Quickly. Considering that there is incomplete dominance still. Now this is where interesting thing comes up. I want the answers in the chat. Again, we are self-crossing F1 to get F2. What will be the genotypic and phenotypic ratio? And this is the question that has come in entrance. So these problems comes as word problems, okay? So when we will discussing the previous year's questions for this topic, we will come to that. But let's practice first. Okay, I've received one answer. Madhya, would you like to just cross check? I know that you are understanding, but you just made one silly mistake. Just one. Very good, Saifullah. Minashi, remember the word incomplete dominance has to be factored in. You also made just one silly mistake. Don't be in a haste. Be slow when you are learning. Once you have learned, you can just keep on getting faster and faster. Both Madhya and Minashi, I will be glad if you can find that mistake. Yes, Madhya is learning. Beautiful, Madhya. You understood, right, Madhya?
Yes.
Yes. And you are thinking right now, oh, this was easy. Minashi, could you figure out that one silly mistake you did? Okay, Mamuna, everything is clear to you, I believe?
Yes.
Minashi, will you please help me in doing this? Okay, you will, like, I'll follow your lead, okay? The question says, let's start writing in different pain. We are self-fertilizing F1. So tell me what will be parent A's genotype?
Capital T, small t.
Perfect. And B's genotype?
Capital T, small t.
Perfect. Everyone starts correct. And then The next thing is Punnett Square. Are you all making Punnett Square or are you saving time? Don't save time right in the beginning. I hope Minashi, you are making all these squares yourself.
Yes, I made.
Okay, then I think there is some confusion with which will go with practice. Okay, tell me for parent A, what will be the possible gametes?
Capital T, small t.
Perfect. And for this?
Capital T, small t.
Right. So this is F1. In F2, what will be the first progeny genotype?
Capital T, capital T.
Perfect.
Capital T, small t.
Okay.
Capital T, small t.
Okay.
Small t, small t.
Okay. So incomplete dominance do not have any effect on the genotype, right?
Yes.
What is the genotype? What are the different types? One thing is this is possible. What else is possible?
1:1.
Okay, so you're giving me the answer directly. 1:2:1, right?
Yes.
What is this 2 and what is this 1?
Capital T, small t, and
2.
small t, small t.
Okay. This part is clear to you, right, Minashi? And we did it
Yes.
right. Now coming to phenotypic ratio. Remember there is an incomplete dominance.
Yes.
But incomplete dominance works in heterozygous state, right?
Yeah.
So this part here is going to be, what is this going to be? Now, if there is incomplete dominance, what are the possible phenotypes? It can be tall. It can also be medium now. You understand? And then it can be short. Yes or no? Everyone
Yes, sir.
understands this part? So tell me how many will be tall?
1.
Yes, right. Because to be tall now, you have to be pure line. Because the moment you are not pure line, you are not dominant completely. So capital T also has the same dilemma now that small t had. To express themselves completely, they have to be homozygous. So this has to be tall. How many will be medium?
2.
Why? Which one? Which two will be medium? These two will.
Capital T.
And why they will be medium, Minashi?
Was heterozygous.
And there is incomplete dominance. So the capital T is not able to dominate the small t now. And the small t is pulling the height down. So capital T wants to be tall. Small t is saying enough of your bullying. This is not where we are following dominance. So let's settle in between. So let's not be short. Let's not be tall. Let's be medium. Yes. So it will be 2. And what will be the short plant?
1.
So do you all understand that because of incomplete dominance in F2, when we start from pure line parents, the genotypic ratio is same as phenotypic ratio? Is it clear to everyone? Mamuna, Madiha, Minashi? See, this is very important. If you have issues with this, it will be difficult for us to move forward. So let's all be on the same page. Is it correct? Mamuna, is it correct? Anything?
Yes, sir.
Yes, perfect. Now write down this kind of incomplete dominance is shown by snapdragon flower. There is a plant called snapdragon, not in terms of height, but in terms of color. So this one that I talked about, this happens in snapdragon plant. Okay, and the trait in character in which it happens is flower color. Okay, everyone. So I think everyone is understanding what we are doing, right? Madhya, how are you finding it?
Until now, I understood everything.
Yes, that's good. You know, it's just that one step at a time and the genetics will be finished and you're like, oh, this was it. It was simple. Now, should I give you one more question where you will follow? Okay, just write down. If a homozygous short, if a homozygous short plant is crossed with, or write down if a homozygous recessive plant. So I'm talking about height, okay? The character is height. So a homozygous recessive is crossed with a heterozygous plant. Homozygous recessive, the character is given as height. Homozygous recessive is crossed with heterozygous. In parent generation, considering that there is incomplete dominance in this species, now there are three parts of the question. Find out what will be the GR of F1, first part. What will be the PR of F1? And third part, what will be the phenotypes of parent? Question is clear? Madiha, Minashi, Mamuna, and Safla?
Yes.
Please find out and give me the answer quickly. And I'm marking the parents for your ease as A and B. Okay, so you have to tell me the phenotype for A and phenotype for B in the chat. Homozygous recessive is parent A and heterozygous one is parent B. Yes. Madhya, again, you are making a silly mistake. Consider incomplete dominance, please, in your answer. I'll be just back in a moment. Please write your answers in the chat. I'll just get water for myself. Okay, everyone? I'll be just back in 1 minute. Am I audible?
Okay.
Yeah. Okay, everyone, am I visible to you? Am I audible and visible, everyone?
Yes, sir.
Yep. What happened? Everyone is struggling with this? What? See, it's slightly confusing in the beginning. I can understand. I'll give you that margin in the beginning to all of you. But I want all of you to practice. Okay, I received some answers. Okay, this time Mamuna will lead me. Okay, Mamuna, you're there. Please
Yes,
help us with this problem. Okay.
sure.
Okay, everyone. So that it's enough time. Okay, good answers. Okay, fine. So let's see what happens. Okay, so Mamuna, will you please take me to the answer? So what will be the first? Let's follow the same steps. The genotype for A,
Capital B, small b.
capital T,
Yeah, small t.
small t. This,
Yeah,
Mamuna, it's homozygous recessive. Homo.
small t, small t.
Yes. If we would have started with this genotype, after all the hard work, the answer would have been wrong, right? So A is small t, small t. You understand? That's why it is very important to figure out what the parent's genotype is in any before starting any cross. Okay?
Yes,
Yes. Like I know that it was very like silly mistake, but it costs. Okay, B.
capital T and small t.
Yes, perfect. So next, let's make the cross. Mamuna, tell me what will be the gametes possible for A, parent A?
Small t and capital T.
No, parent A.
Small t, sorry.
Yeah. And for parent B?
Capital T and small t.
Yes. So the first progeny?
Capital T, small t.
Okay.
Small
Second.
t, small t.
Okay. Third?
Small t, small t.
Okay. And?
And small t, small t.
Okay. Now let's talk about the genotypic ratio first. Before coming to genotypic ratio, let's analyze here only. If considering law of incomplete dominance, what will be parent A's phenotype? If the genotype is small t, what will be the phenotype? Mamuna. Now there is an incomplete dominance. So the trick is that it will not be binary, either tall or short. There can be medium also, right? That is the only thing that's get added up, right? Mamuna. So what will be
Yes.
A phenotypically? Because this is the third part of your question, phenotype of the parent. Tell me.
I think it will be recessive only, no?
Yes, because incomplete dominance only applies to a dominant trait. Yes.
Yes.
Is there any dominant trait here?
No.
So the plant will be short or dwarf, right?
Yes.
And to just say it more properly, it will be pure line short or a homozygous short. Yes.
Yes.
What will be B's phenotype?
It could be either short or tall.
No. Incomplete dominance.
Okay, then it will be tall.
It will be medium. Because this T here will not be able to show its dominance completely over small t every time. So this will try to make plant tall, but this will pull down the height and make it medium height. You understand? Because in incomplete dominance, there is blending of characters. So what will we get if we blend tall and short?
Medium.
Do you understand, Mamuna?
Yes.
Just some very careful consideration to the question, and it's simple. Incomplete dominance, whenever it comes into picture, it blends the characters. Is it clear, everyone?
Yes.
Yes. Cool. So third part is clear. The answer is pure line short for parent A and medium height for parent B. Okay. Or you can say short or medium for parent A and B. Now let's come to genotypic ratio here. Tell me, Mamuna, what will be the genotypic ratio?
1:3.
Okay. And what are these genotypes? What is 1 and what is 3?
1 is medium and the 3 is short.
Genotype. Medium and short are phenotypes.
Okay, then one is tall.
One is capital T, small t. We are talking
Yeah,
about
okay.
genotype.
Right.
Is 2. What is 3? 3 is
Small t, small t.
This is genotype. Okay. Okay, everyone. Coming to phenotypic ratio, ma'am, what will be the phenotypic ratio?
It will be the same.
What same?
1:3.
Perfect. 1:3. But tell me what will be the phenotypes possible?
Sir, there are two mediums, sir.
There are two mediums.
Yes.
That is capital T and
Third,
small t.
the third box.
The third box is small t and small t. Because parent A is pure line short.
No, sir, the before one.
Before one, sir.
That one, yeah.
Capital T is small t.
Yes, so this will be
Below
medium,
it.
right?
Below it.
This one, no?
Yes.
So it will be, oh, okay. Yes. Right. Made a mistake. Yes, perfect. We both made a mistake, Mamuna.
Yes.
Mamuna is feeling happy. I'm not the only one who make mistakes here. Okay. Yes, slight consideration. Thank you so much, Saifullah, for this. Now tell me what will be the
2:2.
so this will also change now, right?
Yeah.
Yes. Yeah, so genotypic ratio will be 2:2. What will be the genotypes?
Capital T, small t, and small t.
Is 2, small t. Perfect. And what will be the phenotype?
Capital, 2:2.
2:2. And what will be the phenotypes possible?
2 mediums and 2 short.
Yes, there will be no tall, right?
Yes.
2 medium versus 2 medium is to 2 short, right? If the same cross was done without bringing in incomplete dominance, what would have been the change? Just quickly here only. And the question is for Madhya. Madhya, right now, just tell me. If there was no incomplete dominance playing the part and law of dominance was being followed, the same cross, what would be the genotypic ratio and phenotypic ratio? Madhya?
Genotypic ratio would have been 2:2.
Yes, genotypic ratio would have stayed the same even if there is no incomplete dominance. So this proves that incomplete dominance has no effect on genotype, right?
Yes.
We're getting prepared for another question. Incomplete dominance has its effects on phenotype, genotype, both, none.
On phenotype.
Yes, perfect. And what will be the phenotypic ratio, Madhya?
That will also be 2:2.
Yes, but what will be the phenotypes then?
The phenotypes will be tall and short.
Yes, hybrid tall and short, not medium and short. Yes, understood? Perfect. So I think everyone is understanding, right?
Yes.
So when you go back today, okay, you have to, let me give you a homework. You have to read from NCERT, the first 4 pages of chapter 5, where they talk about till codominance. And your homework is to read about codominance. And before codominance, even if you don't read about codominance, so the second exception to the law of dominance is codominance, okay? In exceptions, the first one we did today was incomplete dominance. Write down. The second exception is codominance. What does the word co means? Co-ed school.
I guess both.
Both, right? So codominance means there is no recessive or no dominant trait. That is a complete ignorance of law of dominance. The first scenario that I give you a command, you do not follow it at all, right, Madhya? And this happens in our blood group. The example is ABO blood grouping. So I just want you to go and revise blood group. Even if you don't understand the codominance part of it, just revise that what is A, B, and O. And also when you're revising it, also revise what is RH factor, because you all must have, you all know that your blood is either AB or O, and with positive or negative, right? So what is that positive or negative? That is the RH factor. Yes? Everyone? Namuna? Is it clear?
Yes, sir.
Yep. So your homework is you have to read about ABO blood grouping. This, even if you don't understand the codominance part of it, it's okay. We'll do it in next class, and we'll finish other laws also before we go to recombination and some other higher concepts. So I will not start ABO grouping because we have just 5 minutes left. So I'll take doubts if anyone has doubts from any chapter, like from previous chapter or this chapter. Any doubt? I see that every one of you in the beginning struggled in some or the other problem, including me myself. Like I didn't struggle, but I just overlooked this capital T here, and the answer was coming to be wrong, which is another very silly mistake we all can do, even if we know the concepts. So we have to keep our brain, eyes both open with hand-eye coordination when we are doing these crosses. Over time, you will not need to do these crosses, okay? I'll tell you shorthand trick to do that, but I did not tell that trick in the beginning because it's also present in books or in internet, but that makes things more confusing. But please give some time for revising these things. Look at every Punnett Square, every GR and PR that we did today. It is the most conceptual chapter that you can ever find in biology, genetics. Okay. Madhya, you are understanding? You are on the same page with me?
Yes.
Till now, do you find any difficulty in genetics?
No, I don't.
Don't miss classes and you will not find difficulty. Okay. If you'll miss classes, there will be a break in the chain of learning. Okay. Minashi, Mamuna, any doubts? Anyone? Any doubts?
Yes, sir.
If there is no doubt, then